Alright, so we're going to be starting our life science unit off with a lesson that deals with DNA, because you really can't understand a lot of the life science standards without first understanding how fundamental DNA is to all living things. It's found in all of our cells, it's found in all of the cells of all living things, minus bacteria and some other very simple organisms that have RNA, which basically functions the same way. But DNA is the sort of codes for life on Earth. And so the first lesson is going to have students actually extracting DNA from a strawberry. And the important things, as the classroom teacher for this one, the important things are going to be to just really try to encourage students to think about how amazing it is that at the end of this lesson they're actually going to have a vial full of DNA, which is it's just incredible, because it's what makes them who they are, and it's what makes them different than the strawberry that they're going to mash up to get the DNA from, and it's what makes them different than all other living things on Earth. Now this lesson is, you can find it on the teacher's guide. The lesson's title is It's Alive, and the standards that we're covering are 8.3.1, students will investigate and describe the structure and function of living organisms. And 8.3.2, students will investigate and describe the relationship between reproduction and heredity. So there's also some key vocabulary that you could encourage students to look at and think about, and that you can find on the teacher's guide located just below this video also. So with that said, let me just walk you through how this lesson is going to work. And so what we'll be doing, first of all, is just talking a little bit about DNA, what it is, where it's found in the cell, and some interesting facts about how our DNA relates to the DNA of other living things. And then after that, we'll actually do the DNA extraction. And so what we'll be using is a Ziploc bag, a strawberry, okay, and they can be frozen or fresh. I like to use frozen because they're quite a bit cheaper. We need a small cup, a coffee filter, and a toothpick. And you're also going to need some DNA extraction buffer, which is made of shampoo, water, and a little bit of salt, and some 91% isopropyl rubbing alcohol, which you can buy at Walmart. Now, for our lesson, I've also sent out some little capsules that we're going to collect the DNA in, and some yarn that we're going to use to turn our DNA into a DNA necklace. And if you're interested, if you're not working with me and you're interested in finding these little capsules, I included a link below the video of a website where you can purchase these. Okay, so I'm going to go ahead and show you how this whole process works, and then we'll kind of wrap up with a little discussion. Okay, so the whole process of extracting DNA from a strawberry is pretty simple. The first thing that the students will need to do is just mash up the strawberry. So they're just going to, in the bag itself, just really mash the strawberry into a pulp. And the more smashed up it gets, the better. And you can explain to students that at this point they're not, they're not getting the DNA out of the cells, because they're not destroying the cells themselves. They're just breaking the strawberry, which is a bunch, billions of cells altogether, into smaller, more manageable chunks. Now, as we know, cells are surrounded. Plant cells have a cell wall, but then they're surrounded by a cell membrane. And the DNA, which is in the nucleus of the cells, also has a membrane around it. And so the next step is to add the DNA extraction buffer. And the shampoo in this is actually going to destroy those membranes and release the DNA along with other cell parts into the liquid. Now you can see this strawberry is fairly dry, and so I'm going to add a little more extraction buffer than I would if there was more juice. It doesn't take a lot of extraction buffer, but you do want to make sure that the kids have plenty of liquid in their bag. So I'm probably going to put in maybe 4 or 5 pipettes full. The kids just need to add in enough so that they do have plenty of liquid. Okay. So another thing I always like to tell students at this point is you've got to be a little bit careful here. You don't want to really get crazy with this, because the shampoo will get really bubbly, and those bubbles are going to stop a lot of the DNA as it gets filtered. It's going to get caught up in those bubbles. So just kind of slowly move the strawberry around through the shampoo, through the extraction buffer, And you're breaking the cells apart at this point. You're releasing now the cell parts, including the DNA, into the liquid. Okay, and so just keep moving it around through here. The more chunks they can get out, the better. Okay, but at this point, okay, it is ready to filter. And you can see I like to have them get it kind of down into one corner of the bag here. And then they're going to take the cup and the coffee filter and set up a really simple filter apparatus here. So you got the cup and the filter, and we're going to take the strawberry DNA extraction buffer mix here and put it into the filter as much as we can get in there. This was a fresh strawberry, and so it's not quite as, it was harder to mush up, I should say, than the frozen one. Now, this will filter eventually, and if you have plenty of class time, you can allow it to filter. But in the short time I have to work with students, I like to have them kind of help the process along. So I have them take the filter and really, really gently apply a little bit of pressure to the filter itself, just so that the juice comes out. Now at this point they've got to be a little careful because they don't want the filter to break. So it's kind of a balancing act between getting as much liquid as they can and getting no chunks of strawberry. Okay, so that's probably enough. And the handy thing about this with the bag is that this can go back in the bag, all right, and then this can act as sort of a place to put our trash. All right, so in here now we have this liquid, and in the liquid is all of the cell parts, many of them broken down because they have a cell membrane, like the mitochondria, the chloroplast, all of those, the membranes would be destroyed. And so, but you also destroyed the nuclear membrane, and so the DNA is in this liquid. Now to get the DNA separated from the mixture, we're going to use our rubbing alcohol. And I usually have kids put about 3 pipettes full of the rubbing alcohol into the liquid here. And what this does is it causes the DNA to precipitate out of the solution. So the DNA now is more of kind of a jelly-like substance. And the last step is to take a toothpick and I don't want the kids to stir it, but twirl it between their finger and their thumb. And when they do that, okay, the DNA should wrap around the end of the toothpick, and when they pull the toothpick out, they're going to have DNA on the end of it. Now this is where you, as the teacher, I would really like you to kind of encourage kids to think about how cool this is. That this slime here, this gross, I mean, it's just slime, basically. I mean, look at this. Ugh, can't hardly get this to stay on. Okay, so this stuff right here, this goo, this stuff right there, that is strawberry DNA. And it's the exact same molecule that they find in all of their cells. Now this obviously is not the double helix shape that we think of when we think of DNA, but it's the same stuff. It's just millions and millions of bits of DNA all mashed together. So I have students then put that goo, that DNA, into their vial here, okay, and I maybe get a little more if I can here. Okay. Now if students don't get any at all, there's a couple of things that they could have done wrong. They could have added the rubbing alcohol before they filtered it. Ooh, that's quite a bit there. Or they maybe need to add a little bit more rubbing alcohol to their mixture. Sometimes that brings some of that DNA out. Okay, so there's my DNA in my vial. And I'm going to add a little bit more rubbing alcohol to this. Okay. And then what I'll do is I'll take this little centrifuge capsule, and I'll take the yarn that I've tied into a loop and close this around. And now kids have this, all right, their very own strawberry DNA necklace that they can wear and remember, you know, that this is DNA, right? This is maybe the most powerful molecule on Earth when it comes to life on Earth. It's what drives all life, okay, DNA. Okay, so students have now extracted their DNA, and we'll kind of get things cleaned up, and we'll end up with a little discussion again about DNA and the power of this molecule. And this is going to be our theme for the rest of our life science unit, is DNA and how it allows animals and actually species to change over time, to evolve as this DNA changes from one generation to the next. And so we're going to slowly work our way towards that. As a follow-up to this lesson, You could have students bring in all kinds of fruits and vegetables and get a couple of blenders and blend things up and really try to extract DNA from other fruits and vegetables, maybe even some hamburger, maybe their own saliva or their dog's saliva, and see if they could get DNA from those sources and compare it. How does it look? How does it compare? How much did you get? You can make this more scientific by sort of measuring, weighing the items that you're going to be extracting DNA from and comparing the amount of DNA you get based on weight. Because a strawberry, for instance, has 8 copies of all of its DNA. So there's a lot of DNA in a strawberry compared to even, say, the same amount of, if you had the same amount of mass of saliva or some other thing from a human, hair, for instance, because we only have 2 copies of all of our DNA. And so it's an interesting experiment, I think, and something that I just want to start with to really get kids thinking about DNA, because that's, again, going to be our focus for the life science unit. Okay, we'll see you next time.